Today I'm talking with Ben Stear, a bioinformatics scientist at the Children's Hospital of Philadelphia, or CHOP. He's also working on a PhD at Drexel University, focusing on how to detect pleiotropy using graph machine learning and graph analysis. His recent work has focused on large data integration and analysis using knowledge graphs, which is mostly what we're going to talk about today. So Ben, can you tell us a little bit more about yourself and what you currently work on?
Sure, yeah. So like you just mentioned, I work at the Children's Hospital of Philadelphia. I've been here for about 7 years. I've been full-time for about 5.5. I started as an intern after I graduated from Drexel. Um, and like you also mentioned, I'm currently getting or working toward getting a PhD at Drexel. Um, where I'm focusing on graphs and network biology and hoping to do a little bit of graph neural network analysis as part of my thesis.
So the reason why I'm really excited to talk more about the kinds of work that you're doing is, I always feel like we're generating, more and more amounts of data, right? I teach a bioinformatics class, and there's this graph where we just sort of look at the overwhelming amount of data that we're generating, whether that's genomics, imaging, clinical data. Um, and for a lot of my students, it feels like, drinking from a fire hose. But the challenge isn't just collecting all of this data, but it's making sense of this tangled web of interconnection. And tools that can really capture how interconnected biology truly is would really help us see more of this bigger picture instead of, kind of just focusing on these little snapshots. And so what I really want to dive into is, the paper that you've published around the Petagraph, which was recently published in the journal Scientific Data. And it's about this large scale unification knowledge graph framework for integrating biomolecular and biomedical knowledge. But first, I should probably back up a little bit and have you actually describe what knowledge graphs are for our listeners. Like what are they and why are they so useful in biomedical research?
Sure, yeah. Knowledge graphs are a type of graph. So graphs are just uh, node and edge representations, basically. So you have what we sometimes will call entities or concepts, and those will be your nodes, and then you'll have edges between the nodes, which represent relationships. And you can go as simple as you want to as complex as you want, where a simple knowledge graph would be, uh, for example, just a bunch of drugs and their interconnections. Or you can have things like heterogeneous knowledge graphs where you have multiple different types of concepts and multiple different types of edges, um, where you have things like genes and pathways and their interactions and um, variants and all, whatever you can imagine, really. Um, and knowledge graphs are really well suited for modeling data that contains lots of relationships. And as we know in biology, biology can be extremely complex, um, where you have, like you mentioned, all this type of data, where you can have genomic data and epigenetic data, and you can have metadata about the patient, and all of these different types of features. And graphs and knowledge graphs are really useful for modeling this type of thing.
So when I think about knowledge graphs, I kind of think about them as maybe, you're looking at a map and there's a million different modes and routes to get to a particular place you want to go to. And a knowledge graph is, creating the most direct or most meaningful route to get to, maybe a new store, new restaurant, something like that, right? This idea that a knowledge graph really helps you to traverse a particular path in a particular mode that you want. Do you think that's a decent analogy, or do you have a kind of an analogy you think about when you think about knowledge graphs?
Yeah, sure. So you can definitely think about it that way. That's sort of I would say one application, sort of like a finding the shortest path between two concepts, is sort of like an application you can apply on a knowledge graph. But there's just tons and tons of different applications or use cases, I guess you could say, for knowledge graphs where you can do things like traditional machine learning with subgraphs, where you will pull some subgraph or some piece of the graph out and do like traditional analysis for machine learning on it. Or you can do things like apply like graph neural networks, where you apply predictive learning algorithms directly on the graph structure as it is. And there's tons and tons of different graph algorithms and network analysis methods that can be applied to graphs. So traversing the graph and going from one point to another, I would say is a specific type of application within the wider graph algorithm and graph analysis field, if that makes sense.
No, it does. It makes perfect sense. And to be fair, I'm definitely coming at this from a genomics lens. So my most of my interaction with knowledge graphs originally stem from like graph-based genomics, where you really are trying to traverse a specific set of paths.
Yeah.
But thank you for that, expanding that out a little bit more for me. Speaking of, you mentioned lots of different kinds of graphs. What inspired the creation of Petagraph specifically, and how does it differ from other biomedical knowledge graphs?
Yeah, that's a great question because there are at least, I think, three or four, um, at least in recent years, very large scale knowledge graphs like Petagraph. So I'll start with what inspired the project and how we kind of got started. So my PI here at CHOP, Deanne Taylor, uh, had noticed, like a lot of other people, the scale of biomedical data that's coming out of these machines, right? Each year, it seems like it gets cheaper and cheaper to generate more and more data. And also, each year, it seems like there's uh, new methods to generate different kinds of data. All the different types of uh, genomic sequencing methods and transcriptomic methods, and all of that. So there's more and more data, and there's more and more different kinds of data basically coming out each year. And so we wanted a way to kind of take advantage of this on a larger scale. And so that's when we started getting interested in putting together a type of knowledge graph. And so while we were kind of doing a bit of literature search, seeing what was out there, we came across a researcher in Pittsburgh named Jonathan Silverstein, who was sort of already doing this. And what he was doing was creating a property graph instance of something called the UMLS, which is the Unified Medical Language System, which is a giant corpus of knowledge in graph format that standardizes a bunch of different biomedical concepts. So what Jonathan did in his spare time, kind of as a side project, implemented this UMLS structure into a property graph using a platform called Neo4j. So Neo4j is a platform where you can create knowledge graphs and interact with them. And so he kind of got this thing rolling on his own by working on this. So we uh, contacted him and eventually started working with him, and that's kind of how the process of Petagraph was born.
That's a really cool story. But I did want to ask, so you've mentioned property graphs a couple of times now. So what's the difference between property graph versus knowledge graphs?
Um, so I think they're pretty closely related. When I think of property graph, I just think of The nodes can have their own properties, and edges actually as well can have their own properties. There's other types of graphs where that's not the case, and I guess a knowledge graph uh, will always be a property graph. I'm not sure of the exact semantics there, but when I say property graph, I'm just speaking of graphs that can have properties on their nodes and edges, and Petagraph is a property graph.
Gotcha. Thank you for that clarification. That's not a term I've come across as often as just like knowledge graphs in general. So I appreciate that.
Sure.
You mentioned that you're talking about really large scale for Petagraph. So what is that scale? Like how much data are we talking about that you utilized for creating this?
Mhm. So the I believe that at the time of publication, we had there's about 30 million nodes in the graph and something like 150 or 160 million relationships. And it sounds like a lot. It is a lot, but I will say that we have probably five or six different data sources that make up huge percentage of that. So, I mean, it is a lot of data, but um, there's probably five or six different data sources that make up the lion's share of the concepts and edge counts there. Um, but we do have about 180 or so different data sources. Now, about 100 and I think 15, about 115 of them come directly from the UMLS. And so we build on top of the UMLS, I think, which we'll talk about later, um, is called the UBKG, but we'll get into a bit of that later.
So, we've talked a bit about different kinds of data. So, for instance, let's say we've got information about genes, how those genes are expressed, what kind of proteins they make, clinical histories involving those things. All of them are collected differently, right? Different technologies, different formats, often described using different terms. So I'd like to talk a little bit about like how does one even unify all of those different data types? How do you handle that ontological unification? Um, and how are these ontologies essential in, those building blocks for organizing and linking data into knowledge graphs?
Sure. Yeah, so I want to make a distinction here between ontologies and standardized vocabularies, and then also actual like quantitative data, like genomic data or transcriptomics data, whatever it is, because we started with um, unifying these ontologies and standardized vocabularies. Um, so, for example, things like HGNC or ensemble, which are different standardized ways of naming genes, right? Um, and things like Uberon, which is an anatomy ontology. And these things are what we call ontologies or standardized vocabularies, and they're really just structured ways of organizing data. They're often hierarchical. So, for example, in the Uberon anatomy ontology, you'll have something like a very high level body system or tissue like heart, and then you'll get into the sub, the sub tissues of the heart, different parts of it, and you'll get break it down all the way to the molecular level. So those are ontologies or standardized vocabularies, and we started out uh, integrating just those. So originally we didn't have a ton of actual quantitative data. We focused on the ontologies first. And to answer your question, how we went about unifying them is that we met uh, twice a week with Jonathan in Pittsburgh and his team of developers, and really just talked it out about the best ways to introduce new ontologies piece by piece. So we would kind of look around and decide together what made the most sense to ingest into the graph or add to the graph. And we would talk about what links do we want these nodes, these new nodes to have. What's the best way to incorporate them into the graph? And so we kind of just took it one data set at a time like that. Um, and yeah, that's how we kind of slowly worked our way up to where we are now over the course of a couple of years.
That sounds like a lot of work.
Hmm.
What were some of the challenges of trying to unify so many different ontologies, and how do you balance standardization with like flexibility for trying to incorporate future data sets?
Yeah, it's definitely a challenge. I'd say one of the biggest challenges is just being consistent with how we go about integrating data and how we go about modeling data. Um, because you can imagine doing this over a long period of time. You want to know what you ingested. Sorry, I use the word ingested sometimes. It's kind of like a buzzword for adding new data to the graph or adding new data to a database. But so it's easy to add data at one time, but if you want to be doing this over multi-year time frame, you need to document things and be very organized with how you go about modeling these new data types you're adding to the graph, just to make sure you're consistent. Because when you have this scale of data and this many different types of uh, whether it be ontologies or actual data, you want to be consistent with how you're modeling them. And so what we did was, like I said, we met twice a week, and there's a lot of communication going on behind the scenes with myself and my team members, and then also just um, documenting things rigorously.
Which is always the secret to any good project, right?
Yes,
Documentation.
exactly.
Um, so in the paper, I really loved this phrase you kind of used, and you said that you're bringing the data to the ontologies. What does that actually mean, though, in practice for you all?
Hmm. Yeah, I love that phrase too. That was something our PI Dan had kind of coined, and it's simple. It just means that we started out by unifying these different ontologies. Like I mentioned, the Uberon ontology for anatomy and the different standardized vocabularies like HGNC and ensemble, um, and things like the human phenotype ontology or the mammalian phenotype ontology for mouse models. All of these different ontologies, we built and unified them first. And then what we did was we started integrating actual genomics or transcriptomics or epigenomics data sets. So it's a very semantically rich kind of backbone we have here. And then when you lay quantitative data on top of that, it's it allows for much more meaningful analysis or exploration. So that's what we mean by bringing the data to the ontologies.
And that quantitative data, does it just end up translating into weights, or how is that actually handled within the knowledge graph?
Yeah, we use a couple different methods. So um, it can be tricky when you start incorporating quantitative data, because you have to create a node, or you have to add that quantitative value as a property somewhere. So one thing we've done is, for example, the uh, genotype tissue expression database, or GTEx. We created a GTEx node, which sort of, if you think about it at first glance, doesn't make a lot of sense. It's a GTEx node that's doesn't really, have any concrete meaning. It's just the value of a gene in a tissue. But what we did was we connected the tissue nodes from Uberon and the gene nodes from HGNC, and we stuck a node right in the middle of them. So this GTEx node will have a connection to a tissue and a connection to the respective gene. Um, and then we basically binned uh, the GTEx values between, we just created a uniform binning system. So we didn't include the exact expression value. We just created
Ah, okay,
these
okay. That makes
binned
sense.
nodes. Yeah, so we did it that way to save a lot of space, but also, um, we wanted to do it this way to stick to the schema that we inherited from the UMLS and how Jonathan had put his Neo4j graph together. And we can talk about this schema a little bit later if you want as well. But that's one of the things we had to consider when integrating new data is how rigorously do we want to stick to the schema.
I do actually want to talk a little bit more about the Unified Biomedical Knowledge Graph. So you had mentioned that it was this foundation, if I'm remembering correctly, for Petagraph.
Yep.
So can you talk a little bit more about what that is and why it was such a good foundation?
Sure. Yeah. The Unified Biomedical Knowledge Graph is just the name we've given to the uh, the graph form of the UMLS. So just to state again, the UMLS is this huge collection of ontologies and standardized biomedical vocabularies organized uh, by NIH, and our colleague over at Pittsburgh had put all of this into a Neo4j graph instance. So he turned it into a graph. Um, it did exist as sets of triples. So you can think of a graph as a giant set of triples, basically, where you have subject, predicate, object. So you have the two nodes, the subject and the object, and then the predicate is the relationship between them. That's sort of how the UMLS existed prior to Jonathan turning it into a actual graph where you can go and interact with it using Neo4j. But that's what we uh, call, or we named that the Unified Biomedical Knowledge Graph. And then to build Petagraph, we added a bunch of data on top of that.
Ah, okay, gotcha, gotcha. Thank
Yeah,
you for clarifying that. Um, so how many what kinds of new relationships or linkages does Petagraph introduce as opposed to these other ones we've discussed?
sure. So Petagraph adds basically all of the major ontologies and uh, standardized vocabularies that we could think to add that the UMLS didn't include. So that was sort of our first course of action. All of these um, human phenotype ontology and mammalian ontologies, things like I'd already mentioned, Uberon, the anatomy ontology. Um, we added things like um, NCI data or data for variants. And then we also put in um, quantitative data. So things like, one second, things like hub map, so all sorts of different transcriptomics and genomics data, things like variants, and their population frequencies. We wanted to focus on congenital heart defects for a specific use case. So we have a bunch of um, RNA sequencing data from the developing heart. Um, but there's just a ton of data we've put in there.
I think this is a great point to talk a little bit more about some of the potential real world examples about how researchers might use Petagraph. So you'd mentioned the heart a little bit, but I know in the paper you talked about like disease gene prioritization, integrating multi-omics data. But is there one use case in particular you talked about in the paper that like you're really excited about that you could walk us through a little bit more?
Sure, yeah. It's really cool because Petagraph differs from some of the other really large scale knowledge graphs in that it is a very general graph. So if you want to do something specific, you might need to incorporate some of your own data, and we have scripts and automated ways of doing that, which make it relatively easy. But there are plenty of use cases that can be done directly on the current version of the graph. So things like disease gene relationship detection or gene disease link prediction can be done. Same with gene disease variant um, prediction. We also have drug targets and some drug properties in the graph, and they've been connected to the known phenotypes and diseases that they affect and treat. So anything you can think of really can be done. One of the more interesting, or something that I at least find more interesting that's not in the paper, is what I'll be doing my PhD project on, which is using some of the regulatory network relationships. So things like promoters and enhancers, EQTLs, and things like that are great for uh, being modeled in a graph because they have relationships with variants and genes, and you can do a lot of fun things in that vein. But for my PhD project, which I'm kind of excited about, I'll be using graphs and taking advantage of these regulatory networks to study the shared genetic risk between two diseases. So it's easy to sort of as a first, simplest use case to just pull out all the genes that are shared or the variants that are shared between two different diseases, and kind of see what the most common EQTLs are between two diseases. So studying things like that has excited me, and I'll be developing ways on how to hopefully do that and also validate that.
So now I'm curious, is your interest in knowledge graphs driven because of your sort of biological questions you're trying to answer, or is it flipped? Were you always kind of interested in knowledge graphs and then you found this particular use case that got you really excited?
Uh, I would say a little of both, actually. But doing this project and sort of following Deanne's lead, she sort of had a vision for integrating this data and taking advantage of the data because once you can organize and integrate the data in a intelligent way, you can take advantage of it, right? You can sort of capture a model of the data that's more closely aligned with reality than, say, just looking at gene expression profiles alone or just looking at epigenomic profiles alone. You can sort of combine all of this and use analysis or machine learning to study something that is not quite reality, but maybe more closely resembles reality than just studying one modality at a time. Um, so that sort of got me excited because a great way to do that is to use graphs and use um, knowledge graphs basically to model um, disease and to model biology. So I was on board when we started this journey into graphs, and we've been doing it for a little while now, a few years. So I've definitely started seeing things um, as a use case for graphs more than I had in the past. Um, but yeah, I'm excited to study these shared genetic risks between any two different diseases using this kind of model.
So you just talked a little bit about, your PhD project and how cool it is and how you're going to, work with knowledge graphs and to answer some really interesting questions. So I'd be really curious to hear on, how you think about the value of focusing on interpreted and curated data rather than, raw experimental output alone.
Yeah, it's a great question, and it makes me think of how our colleague in Pittsburgh, Jonathan, goes about talking about this stuff and how he makes decisions on how to integrate data because it is super important in how you integrate curated process data versus raw raw data because you can think, there's an infinite number of ways to process data, especially when you start getting involved with multi-step pipelines. And if you've ever done it before, you know how many parameters go into it, how many different input files go into it at each
Each
step.
tool has a million options.
Right. Exactly. So it can be hard to reproduce things, when you're working with the same data set. So when you start integrating multiple data sets, you have to really be careful because it can be tricky or impossible to compare or synthesize two data sets that have been processed in different ways. Um, so it's always nice to incorporate data that's been sort of summarized or even just processed and filtered. Um, but the flip side of that is you have to be very diligent about how you integrate that data. I mean, even just you can think of just uh, cutting off or using p-value thresholds. If you use two different p-value thresholds on two different data sets, and then you want to compare the two in your graph or integrate both of them, it might not make sense. So it's nice to incorporate processed or summarized data, and we've done that when we see fit that it works. Um, but also on the flip side, working with raw data, it raw data sort of is what you get, and you can let the user decide how they want to process things. So there's pros and cons to doing both.
That's a fair point. It's almost like summarized data, but not necessarily further filtered, right? Like it
Yeah,
would be
exactly.
easier to use, for instance, expression data that people haven't already filtered out for twofold or something like that.
Exactly.
Okay, that's a good point. Yeah. So I definitely want to ask a little bit about what role can Petagraph play in machine learning or predictive modeling? Is that kind of a direction that it can take?
Yeah, absolutely. And that's one of our end goals here. And something we've already started doing is using machine learning models to uh, predict and analyze the graph. So when you talk about graphs and machine learning on graphs, I think I already mentioned that you can do some, more traditional machine learning models, um, uh, which I basically consider everything except graph. machine learning, which I don't know if that's legit or not, but I sort of separate the two based on whether you're applying uh, uh, neural networks or machine learning that can directly work with the graph structure versus all the other machine learning models. But both, both are super useful, obviously. Um, and yeah, I think Petagraph is super can be used in just an infinite number of ways here to predict and classify and do whatever you can think of. You can do things like link prediction, where you are basically predicting links between different concepts in the graph, whether it's uh, drug repurposing for diseases, or it's specifying or predicting genes and variants to be associated with disease. Or you can do things like classification, where you can have, if you introduce patient data, which we've started doing, um, which you have to be careful about, obviously, but you can have control patients and case patients, where you can train models to pick up disease profiles and then predict on new uh, a new group of patients whether or not they are at risk for the disease. So there's just tons and tons of things that can be done here, and we hope that people will take advantage of it.
So I'm curious, how is Petagraph currently used in the NIH Common Fund Data Ecosystems Data Distillery project?
Yeah, so Petagraph is different than Data Distillery. Data Distillery builds off of the same foundation that Petagraph does, the UBKG graph, but they are distinct graphs. Data Distillery uses data strictly from um, the Common Fund, uh, the different data resource centers within the Common Fund. Um, and so that what the data distillery graph project entails is each of the data centers have sort of submitted what they think is the best kind of summarized data from their projects, and then we've integrated that into what we call the data distillery graph. So both Petagraph and data distillery graphs are built on top of the UBKG graph, but they are sort of distinct.
Gotcha. Okay.
Yep.
Um, so I'd love to ask, what advice would you give to researchers or developers who want to build or contribute to, Petagraph or, even jump into biomedical knowledge graphs?
Yeah. So if you want to get involved or even just use and check out the graph, um, you can just read the paper or go to our GitHub. We try to make it as easy as possible to build a local instance. So it is nice. We have a ton of data in the graph, but it's actually still small enough to be run locally on your laptop. You just need to download the Neo4j desktop application, and then download the graph files that we make available for free. All you need to do is get a UMLS license, which is easy. It just, usually takes less than a day. Um, so that's how you can get involved or build onto the knowledge graph however you want. Um, and then for the second part of your question, getting involved with knowledge graphs in general, it can be a little daunting. At least it was for me when we first started. We didn't really, or I didn't really have any idea on how to actually get this data into graph format. So that was sort of the first big hurdle. But Neo4j makes it super easy. You can basically just have an Excel spreadsheet with three columns, which represent your edges. So it'll be like I mentioned earlier, just the triples where you have subject, predicate, object. And Neo4j makes it really easy. They have a set of standardized tools where you can ingest these really large scale data sets quickly and easily. So I would definitely check out Neo4j and their documentation. They also have a bunch of built-in graph algorithms and tools for visualization and everything that you'll need to get started.
Awesome. Thank you so much for that. And we'll definitely have a link to the paper and the GitHub that you can find. So I did want to end with sort of a fun question. So you'd mentioned we've talked a little bit about gene data, and you talked about handling EQTLs and enhancers and things like that. So I'm hoping that you can answer my fun question, which is, do you have a favorite gene?
Oh boy. You know, I when I was when I first started at Chop, I was helping this guy named Steve Pastor work on a project that was involved with uh, a disease called 22q deletion syndrome. And I don't remember the name of the gene, but it was so kind of a letdown. But basically, the gene was involved in destabilizing uh, breakpoints or something, where basically, if this gene was had pathogenic mutation, it uh, you know increased the chances of something like chromothripsis, I think it's called, where basically your chromosomes are just shattered and it's disastrous. And I thought, wow, that sounds like a very important gene. But other than that, I don't know. I don't really have a favorite gene. TP53 is usually the one that I enter into databases when they have a keyword search. When I want to figure out how a database works, I'll just slide that one in there because it comes to mind.
That's fair. Although I love that your answer basically was like a little mini like knowledge graph. It was like function to like structure and things like that. So that was great. Well, I do want to say thank you so much for joining us today and sharing a little bit more about knowledge graphs and specifically your publication in this space. And hopefully our listeners will be really excited to go and read more about it. So thank you so much, Ben, for your time and have a great day.
Thank you so much. Have a great day, guys.
You have been listening to Decoding the Data Ecosystem, a CFDE Training Center podcast. Be sure to check out our other episodes in this series. Find out more information about CFDE data and tools that may be valuable to your research by visiting our website.